Yeah, there's some papers that just recently came out about the psilocybin gene cluster, and they're figuring out how they all evolved and everything. Those aren't the genes that I look at. The genes that I look at is the junk DNA that does absolutely nothing, because that's the barcode genes that are used to identify things. So it's the same sort of PCR Sanger sequencing that you'll do with anything else to identify these psilocybin mushrooms.
Hi everybody, welcome back. So we're highlighting community members in the Biohacking and Genetic Engineering Design Group on Facebook. So I'd love to welcome Alan Rockefeller. Hi.
Hi, Esther.
What's going on, Alan?
I'm here at home working on a whole bunch of science stuff today.
Science stuff, groovy. I'd love to just tell a little bit about Alan. So I actually met him through work. So him and I worked at the Odin early in the days where we were packaging kits and setting out genetic engineering CRISPR kits. So it's always a pleasure to speak to him about mushrooms, especially. And something that I would love to tell you about him is he has sequenced over 500 fungi in GenBank. So that is incredible in itself. I think Alan might be the biggest mushroom expert that I know, and it's such a big deal to have him on here. So welcome.
Thank you.
Yeah. So I want to kind of get to know who you are, like kind of from the beginning of just like, how did you get started in mushrooms and, the exploration of genetics of mushrooms? Let's start there.
I've always been into nature, but about 2001, I went hiking on Christmas Day, and there was like mushrooms everywhere. I was down in Redwood City, and so I decided I wanted to figure out like which ones were cool and which ones were rare and interesting, and so I just started paying attention to them when I was just out hiking.
Okay. So did you at that time, like what was when you made this transition of like, ooh, I really like just observing mushrooms. Like how do how do I learn more about them? Like what was that kind of spark or that moment that you decided that you wanted to become a scientist in fungus?
I mean, that was about 2001. It was kind of hard. There weren't a lot of internet resources. So I started out by editing a lot of Wikipedia articles. And in doing research for those Wikipedia articles, I learned a lot and kind of dug into all of the papers. And then in 2007, I started going to Mexico for mushrooms. And I was pretty hooked by then. And then in 2012, I quit my office job just to do mushroom stuff full time.
So what's mushroom stuff for you when you go to Mexico?
I spend a lot of time outside, just observing in the woods, looking for mushrooms, and then I'll take the best pictures I can of them, and then I will bring them home. So I collect everything, I photograph, bring it home, and do microscopy and DNA sequencing.
Okay. So with that, you've amassed this incredible amount of 500 sequenced fungi and GenBank, like I said. Do you want to like explore them just for the sake of, putting them in like a cladogram or trying to discover new mushrooms? Like, how does that work?
Yeah, I mean, all of those things. And, it's also just for myself. I see something out in the woods and I'm really curious about it. So maybe I'll pull out my phone and see if there's a sequence of it in GenBank. And if there is a sequence of it, I'll blast it and see what it's closely related to. And then I'll look at it and be like, oh, yeah, it really does fit in there. And then I'll just understand it better. But sometimes there isn't a sequence of it. So then I'll sequence it and be the first person to see where it fits into everything and upload that data to GenBank. So then, when other people wonder where something fits in, they will pull up my sequences and use them. And so they've been cited in lots of papers and stuff that way. And it's pretty good for discovering new species because, in California, we use a lot of European names. Mm-hmm. So I'll just pull out my phone, open up the browser, go to GenBank and pull up some European sequences and blast those and see how they compare to California sequences. And, sometimes they match. And then I'm like, oh, okay, cool. This European name works in California. And sometimes they're way different. And then I'll look around for California names, something that maybe people think is a synonym, but actually one of them was named from the United States and one from Europe. If I can't find any synonyms or any other things that could apply, then I'll think, oh, this probably doesn't have a name yet.
So is that kind of like the thrill that you're searching for, is to find these new species and hopefully in the future kind of name them yourself?
No, mostly I just try to understand nature.
Uh-huh.
So I notice a lot of new species while I'm out there. In fact, there's a lot of mushrooms that haven't been named. If the season's good, if you're walking around, you know what's been named and what hasn't been, then you could easily discover 50 new species, in a day, just because it's so common to see something that doesn't have a name yet. But one cool thing about mycology is there's really no money in naming mushrooms. So like with plants, there's big money in plants, and most plants have been named already. But no one's going to pay you to name mushrooms. So either they'll get named by university researchers who just have a university job, or amateurs who just think they're interesting and they're interested in a certain group. Or more often they don't get named and they just sit there. And when they come up and I see them online, I'll be like, oh yeah, that one's really cool, and it doesn't have a name yet. And I say that more and more. The more I learn, the more I dig into these sequences, the more common it is. So be like, yeah, that one doesn't have a name either.
So, I mean, we know like how important mushrooms are, especially to the ecosystem, and even humans consume so many mushrooms with different types of properties. Why isn't there more research being done on mushrooms? Because I mean, from what I understand, the genome is so large, and like you said, you find like 50 new mushroom species on your first foray. So why is it so difficult to, have a database for all of this existing already?
Well, I think when people do research, they usually want some sort of return on their investment, like they're trying to make money. And there's no way that naming a mushroom is going to directly make you any money. Because these unnamed mushrooms, they're very common. It's not going to change that to have a name. I won't change any commercial applications or anything like that. So people, there's just nobody whose job is to name these things.
Hmm. And I think I had a conversation with you early on about this one legendary mushroom that you've been chasing for many years. And can you tell us a little bit more about that?
Which one do you mean? There's so many that I'm chasing. This week I'm going after hygroscopy and mycena, but last week it was something else, and it's always.
I think it was either between like a 5 or 7 year chase for this one very, very niche mushroom that wasn't in literature very much, but you actually had a chance to photograph in Mexico.
So there's a couple like that. Uh,
Mhm.
One of them is Psilocybe meridionalis, which has only been found one time in recorded history in a very remote place called Sierra de Cacoma in Jalisco, Mexico. And that one, I went to the herbarium and found the type collection. So the one time that it has been found, they keep it, at a university, and I was able to see it and study the microscopy. And in the notes that were along with it, there were some GPS coordinates scribbled down. And so I know exactly where it was collected. And so every summer I'll go there and see if it's there, but it's never been there. So I haven't been able to photograph it. But one person was able to sequence it and put it in GenBank. So I know it's a real species and it's not the same as some of the other species. So that's Psilocybe meridionalis. Another one that I've been going after is Psilocybe moseri, which was described from Chiapas in 1995. And it turns out that when I was in Ecuador, I found it about a year ago. And it was like this waterfall in Ecuador, and these mushrooms were growing from a mud wall almost behind the waterfall. Really cool place. And so I was able to finally get good photos of that one. No, these days, a lot of what I'm chasing are the glow in the dark mushrooms. And I found one recently in the Crystal Springs watershed. And that's a place that's over in San Mateo County. And this mushroom, it glows really brightly at night. And it does not have a name yet. It's a little tiny white mushroom, but it glows green. And so that was pretty unexpected because most of the glowing mushrooms in California are big and relatively dim. Um, so there's, but there's so many more like, mycenas are just really cool. They're tiny little things that grow on wood. And so I've been getting into mycena a little bit recently, and the hygroscopies are super colorful and they've never been studied in California. So every time I see a hygroscopy, I'll pick it and sequence it.
Uh-huh.
But there's so many things to go after. I also get a lot of mushrooms in the mail. Today I got 32 mushrooms from Amanita, and they're all deadly mushrooms from the Amanita expert Rod Talas.
Mhm.
And so I'm going to all those for his Amanita Ameriverosa paper. And then someone gave me about 20 Cortinarius, and somebody else gave me a couple little rare things. I think I got about 6 or 8 boxes in the mail today, and I can tell just by the weight they all have mushrooms. So I'm going to do a big PCR run tonight.
That's awesome. That's fantastic. Yeah, if you ever get a chance to see some of Alan's work, you're an excellent photographer. Like, I'm not gonna lie, as an amateur photographer myself, I see some of your most like beautiful, beautiful mushroom pictures, or even your just travels, and sometimes the occasional empty food plate. But yeah, you take some phenomenal photos, and they've been uploaded to a database, right? I think, yeah. So can you tell us a little bit more about iNaturalist, maybe?
Yeah, so iNaturalist is a website and also an app for Android and iPhone. And so you can just take a picture of any living thing, whether it's a plant or a mushroom or a bird, and put it on iNaturalist, either by taking the picture with the iNaturalist app or uploading pictures on your laptop. And then people will identify it, and it creates a permanent biodiversity record. So it's a really cool way. You just see a mushroom and you don't know what it is. You just put it on there. And so what I do is I go on there every day, and any mushrooms that are found in the Bay Area, I'll identify them. So it's a few hundred a day, but I go through them pretty quick. So it just takes me a few minutes. But I put names on all the Bay Area mushrooms, and that helps me see what's fruiting where. And maybe I'll see something really rare come across there and go out and see if I can find it. And it helps people like know like what they found and where they found it and when. So that's a pretty cool app. And then there's also a website, Mushroom Observer, which is similar but just for mushrooms. So I put all of my photography on both of those websites.
Awesome. Yeah, as just like an amateur user myself for iNaturalist, I just take pictures of random things on my walk, and I'm like, oh, what is this? And immediately somebody on the other end, like an expert like Alan, will just message me and say, oh, this is, X, Y, or Z. And I'm like, this is insane. There are live people on the other end visually, confirming these mushroom species in front of me. So I really love that app. It's a fantastic app, and Alan is very, very on top of it. Awesome. So I want to also highlight that you are one of the most well-known psilocybin mushroom experts. Is that correct?
Yeah, yeah,
Awesome.
yeah.
So
Studying them
what
for
do
a
you,
while.
yeah, what can you tell us a little bit, just a little bit about what you're studying about them and why?
I study mostly the taxonomy, so like all the different species that are out there. So there's about maybe 50 valid species in psilocybe. I'm working on a new one that somebody found in Florida recently. And then there's another new one that's somebody found in Australia that's supposed to be coming in the mail. We'll see if it actually shows up. Um, but that's just one genus. There's actually 6 genera that contain psilocybin. There's Pinealis and Pluteus and Gymnopolis. So I study all those, and those are all like way more obscure too. About a month ago, I found Pluteus phaeocyanopus, which is the Bay Area's rarest psilocybin mushroom. Has only been found a few times, and when I found it, I knew exactly what it was. I'd never seen it before, but that was really cool to see. That was over in San Mateo.
Can you tell us a little bit about, is there like a gene sequence or a specific genes that target, those hallucinations that people get when they take mushrooms?
Yeah, there's some papers that just recently came out about the psilocybin gene cluster, and they're figuring out how they all evolved and everything.
Mhm.
those aren't the genes that I look at. The genes that I look at is the junk DNA that does absolutely nothing, because that's the barcode genes that are used to identify things.
Mhm.
So it's the same sort of PCR Sanger sequencing that you'll do with anything else to identify these psilocybin mushrooms. The only difference is that I have some FASTA files that I've curated, and I know that all the sequences in there are accurately identified. So if you just blast something, you'll get a lot of accurate IDs and a lot of inaccurate IDs because there's a lot of garbage in there. Whereas if I put it in my curated FASTA files and build a tree, then I see where it fits in, among all the accurate names, and
Yeah.
it's a lot more.
So you've built an actual community around just fungal sequencing and barcoding. Do you want to plug that right now?
Yeah, I started a group called Fungal Sequencing on Facebook. So it's just /group/fungal sequencing, and a place for people to ask questions and post their stuff about fungal sequencing. And then I started another one that nobody really uses except for me, but people are welcome to use it, and that's called Fungal Sequencing Results. So if I just get some result and want to post about it, I'll post it in there. And it's like a lot of stuff, you'll get a sequence back and you won't really publish it in a scientific paper because you haven't done all of the research that needs to be done for a scientific paper, but you want to tell somebody. So it's kind of like a halfway in between step between publishing it in a real paper or just keeping it to yourself.
Can a newbie that has never done any barcode sequencing join this group and learn about how to isolate DNA and then run it through a sequencer?
Yeah, for sure. And I have kind of a pictorial where I'll just go through all the steps of the PCR and sequencing process, and also a video. And also on Counterculture Labs wiki, there's a DNA sequencing page where I wrote down just like instructions on how to do it and the primers you need and the equipment that you need, and pretty much everything that you would need to get going is on that little wiki. So I think if you just Google like Counterculture Labs DNA sequencing, that would come right up.
Cool. I'll definitely drop it in the links below so you could check that out so you could see what kind of goes on in DNA barcoding sequencing. And, it's really important that we do all of this fungal work so we can discover brand new species, and hopefully in the future someone can write about them. So that would be pretty cool. Alan also does live forays. Now, when COVID kind of brushes away, then hopefully we can get out there. And how do you announce these forays?
I usually just post them on like Facebook or Instagram.
Okay, and it's like usually like an hour before, right? So you kind of have to like run there.
Yeah, I usually don't post them too much in advance. I'll just be like, oh, I think it's pretty nice out today. It's been raining. I'll just invite the whole internet, and whoever sees the post and they're within range, they'll show up, and we'll go out there and just see what we can find. And pretty cool. A lot of times there's a lot of people, so there's just, instead of me finding everything, it's like people just handing me mushrooms right and left, and I'll tell them about them.
So is there any last words that you want to tell the group about, getting started in exploring the backyard?
There's so many cool things out there in the backyard. Nature is like infinitely cool, and the closer you look, the more you see. So probably the coolest thing to do is just start looking for mushrooms or plants or, anything, and pay attention to them, take pictures of them, put them the photos up on Mushroom Observer and iNaturalist. And if you think it's really cool and you think it might be unique, then run a DNA extraction, run PCR. You can get everything you need to do fungal barcoding or plant barcoding, whatever, in your house for probably under $1,000. And the sequences cost maybe $8 each to generate them, which is almost free compared to how cool the data you get back is. So I'd like to encourage people to get more into their local nature. And also it's always mushroom season somewhere. So right now, it's kind of ending in the Bay Area, but it's just about to start up in the mountains like Mount Shasta, Yosemite. And after that, it'll be mushroom season in the Midwest, the East Coast, Canada, and Mexico. And after that finishes, it'll be mushroom season in California again.
Alan, you're such a fun guy. It's such a great pleasure chatting with you about fungus. So thank you so much for coming on with me. We really appreciate it.
All right, thanks, Esther.
Yeah, no problem. So I want to just take this time to, I'm going to make sure to have all of the links provided to you in this video down below. So be sure to subscribe and like to this channel. And if you're interested in giving one of these talks and pitch a proposal to me, make sure you give me an email, and I'd love to get in contact with you. And of course, join the Facebook group called Biohacking and Genetic Engineering Design. So thanks so much, and I'll catch you later. Bye.